Let's continue our studies of the beginnings of biological compounds by comparing and contrasting monomers and polymers. In order to function, living cells must include some very large molecules, including protein, nucleic acids, and lipids, each serving a function as unique as its structure. These larger molecules result from combining smaller molecules in a kind of modular approach. As we saw in our first video lesson, the order of progression is from simple to complex. These small molecules we call monomers, from the Greek meaning single part. These monomers are then assembled into larger molecules, macromolecules or polymers, containing many parts. Three of our main types of biomolecules can form polymers. Proteins are built from amino acids, nucleic acids from nucleotides, and polysaccharides from monosaccharides. We refer to a single unit within the polymer as a residue. Of the four major types of biomolecules, lipids are not able to form polymeric molecules due to the lack of common functional groups. We'll look at lipids in greater detail in a later lesson. Some of these macromolecules, or polymers, are informational. That is, the order of the monomers within the larger structure carries information. The best example is that of nucleic acid, which is the polymer associated with DNA and genetic inheritance. We'll see that the order of amino acids in proteins also carries some information, and that sequence is vital to function. Of our three main types of polymers, polysaccharides are non-informational. That is, the content is important for function, but not the sequence or order of the monomers. These macromolecules are too repetitive and do not convey information in the order of their monomers. For the two types of informational polymers, we find that they exhibit directionality. That is to say, the two ends of the polymer are unique, whether we're looking at the N to C terminus in proteins or in the 5' to 3' direction in DNA and RNA. Notice that combining monomers to form bonds within the polymer results in release of water, a process that is reversed by hydrolysis, lysis by hydro, that is by adding water. An excellent example of the importance of residue order in a polymer is in the function of enzymes, which we shall see are biological catalysts that have the power to greatly enhance the rates of chemical reactions. The amino acid sequence of these enzymes is absolutely vital to their catalytic roles. The effect of sequence on polymer function is also prominently illustrated in the case of DNA and RNA. The DNA carries the genetic material, and the RNA carries the code that will be translated into an amino acid sequence in the protein product. We refer to proteins as enzymes, and for some time only protein enzymes were known. Scientists have since discovered that there are catalytic RNA molecules, RNA enzymes or ribozymes, which we will consider later. This has led some to speculate that RNA may have served as the original coding material in the early Earth. We know that it does indeed serve this function in the case of some viruses. However, it's important to keep in mind that the subject of the origin of life on Earth is still under active investigation and highly speculative. For the remainder of our studies, let's carry these questions with us as we review what has been proven regarding biochemical molecules and systems and how they function.